to be hosting this fireside chat with Neil Walker, who's the CEO of ACRS AclarisTx, and Walter Smith, who is the founder of Confluence, which was acquired by AclarisTx for its Connect platform and is also heading BD efforts for the company. So thanks for joining us, Neil, Walter.
Thank you. Pleasure.
To start, it would be great if you could just give a high-level background on AclarisTx and your current pipeline for anyone who might be new to the story.
Sure. So AclarisTx is a clinical stage biotechnology company focused on immunology and inflammation. We have 3 clinical stage assets at the moment that are going through a number of phase 2 studies. And we also have a pretty healthy IND-enabling tox set of assets that will be getting to IND towards the end of this year, early next. And then we are actively repopulating the top of the funnel. We have a pipeline of early stage discovery assets looking at neuroinflammation and a number of other targets. And finally, we have a platform that we're investing in that enables us to look at, identify, and discover new targets and move them along quicker to dev candidate in about half the time.
Great. I guess starting with the Connect platform, to your point, what differentiates it versus conventional small molecule drug discovery approaches? And does it allow you to develop treatments with either unique properties or against difficult to reach targets?
Right. So the platform we have is really a combination of a very experienced staff that have a lot of big pharma experience. For example, our vice president of biology is a co-inventor of tofacitinib or Xeljanz. We have our own proprietary chemical library that lets us interrogate the kinase gene family in a short amount of time. And it's very focused on the part of the kinome that's called the cystinome. These have cysteine near the active site, and it allows us to come up with opportunities to inhibit these enzymes with either covalent or irreversible inhibitors or reversible inhibitors. So it allows us to target what we call hard-to-drug kinase targets in record time.
Got it. Turning to your lead asset pipeline. So we'll first dig into ATI-450, which is an MK2 inhibitor currently being developed for rheumatoid arthritis, hidradenitis suppurativa, and then psoriatic arthritis. To start, I think it's helpful if you just give a quick background on what MK2 is, how you landed on MK2 as a target to go after, and then some of the history with targeting P38 and what you're trying to solve with ATI-450.
Right. So MK2 is a kinase downstream of P38. And P38 is a stress-activated protein kinase, meaning in situations of inflammation or stress, this pathway is activated, which leads to the production of inflammatory cytokines. So the industry was really excited about P38 because it was a small molecule approach to inhibiting this kinase and that could regulate inflammatory cytokines and compete with the biologics that were experiencing a lot of success. But P38 operates at the very top of the funnel and has 70 downstream substrates, only one of which regulates inflammatory cytokines, and that's MK2. So the industry had a lot of dry wells in targeting P38 due to tolerability, safety, and pharmacodynamic issues. It became apparent that MK2 was the target of interest to regulating inflammatory cytokines. So the industry changed focus to that kinase. The issue with that is that it's really a hard target to drug with ATP mimetics or ATP competitive inhibitors. And at that time, that was the only approach to inhibiting this kinase. So a number of companies had approaches that never really ended up in the clinic. We became aware of the P38 MK2 co-crystal and found a way to target the interface of that interaction and lock MK2 in an inactive complex. So I think that the issues that were present with the competitive inhibitor approach was overcome, and we were able to generate ATI-450, which was quite active in a number of preclinical models of chronic inflammation, and also went through a phase 1 and now into phase 2 studies.
Got it. And so last year, you reported positive data from a small proof of concept phase 2A study in rheumatoid arthritis with your MK2 inhibitor. Can you just quickly walk through the top-line efficacy data as well as the biomarker data and how those would typically compare to approved treatments in rheumatoid arthritis?
Right. So ATI-450 was taken forward in a proof of concept study in rheumatoid arthritis. And this was done primarily because this is where the P38 inhibitors failed. And they failed for a couple of reasons. One was around tolerability, but the other was around a low degree of anti-inflammatory activity. And one of the things that you could measure with the P38 inhibitors was a transient effect on the inflammatory biomarker CRP. So the study was really designed to look at the change in CRP levels in RA patients, moderate to severe RA patients, over a 12-week period. Of course, we measured all the clinical outcomes as well. And what we found in the study was that not only was CRP suppressed persistently over the 12 weeks, but we also had significant impact on disease endpoints like swollen and tender joints. And this translated into ACR 20/50/70 scores of 60, 33, and 20%, which puts you in the ballpark of DMARD drugs that are on the market. So we were able to demonstrate that by selectively targeting MK2, you could overcome a lot of the issues that were present in the P38 inhibitors, plus have a fairly robust response in moderate to severe RA patients. Now, along with the clinical endpoints, we also measured pharmacodynamic endpoints, cytokine production, target coverage, et cetera. So we have a very good understanding of dose versus pharmacokinetics versus pharmacodynamic endpoints coming from the study, which really helped us and informed us on how to build and construct the phase 2 study that we're in right now.
Got it. And if I remember correctly, about half of patients in the study, the prior study, had prior experience on TNF-alpha inhibitor. And so with this in mind, how do the baseline characteristics from your phase 2A compare to what we typically see in a rheumatoid arthritis study?
Well, the baseline characteristics in the population we looked at, they had to have moderate to severe RA, they had to be on a stable dose of methotrexate, and they had to have elevated CRP, about 5 milligrams per deciliter. So this is a fairly typical RA study, maybe a little bit different in that we had elevated CRP or in this population above 5. But I think it's a fairly standard protocol to explore new mechanisms in RA. Now, we're carrying that forward into a very similar design for our phase 2 study that was underway right now, where individuals have to have moderate to severe RA. They have to be on a stable dose of methotrexate. And we're also including patients that have prior experience with JAK or biologics, but we're limiting that to 20% of the population.
Got it. And so, as you know, targeting MK2 with ATI-450 leads to inhibition of several key inflammatory cytokines such as TNF-alpha, IL-1 beta, IL-6, IL-8, and then IL-17, which was presented in your R&D day. So of note, there are antibodies approved against many of these targets. How should we think about the effect of ATI-450 on things like TNF-alpha, IL-1 beta, IL-6, and IL-17 versus efficacious doses with approved therapies against these targets? And then also if there's any synergies of hitting them all at once that you think also come into play?
I think one of the attractive features of this program is that we are knocking out multiple legs of the stool. So we are impacting a number of cytokines that you mentioned. Also, it's interesting to look at which biologics are active and which have activity in which diseases. So things like anti-TNFs and anti-IL-6s have activity in RA population, in psoriasis and psoriatic arthritis. Anti-TNFs and IL-17s provide benefit. So we're able to modulate a number of different cytokines, and I think that the degree which we can titrate cytokine inhibition has quite a dynamic range. So, of course, biologics are going to knock out a single cytokine at greater than 95 to 99%. We're able to, with our 50 milligram twice a day dose, inhibit at the Cmax over 90% of these inflammatory cytokines. So we're operating even at Cmin at multiples of the IC80. So I think one of the unique features of this approach is A, we're knocking out multiple cytokines, and B, we could titrate the impact on those cytokines to determine what the optimal dose is in each patient population.
Yeah, I think one of the things we tend to mention that some overlook is that, the numbers Wally's talking about relate to the production of these cytokines. We also have an effect on signaling. And so it's hard to say an exact number, but we know it's north of what we talk about because we affect both of those aspects. And it's hard to capture that signaling aspect by just doing an assay.
Got it. That's helpful. I guess going to the assay, so your data shows an impact of ATI-450 on both endogenous plasma cytokine levels and cytokine production in ex vivo LPS-stimulated assays. Can you just walk us through how you think about the relevance of each of these measures and the historical translatability of the ex vivo LPS-stimulated assay towards efficacy?
Right. So we employed a number of pharmacodynamic biomarkers in both the phase 1 and the phase 2 study with ATI-450. And primarily what we were trying to determine was the target coverage versus cytokine inhibition. In the various dose ranges we looked at in phase 1 and also in the patient population in phase 2. So I think what the ex vivo LPS assay allows you to do is have a robust signal where you can look at your plasma level of drug and you can look at the target coverage as far as how much of the enzyme are you inhibiting and what is that level of inhibition, how does that translate into inhibition of cytokine production. So we can connect the dots between those parameters, looking at plasma level to target coverage to cytokine inhibition. We also, in the phase 2 study, looked at endogenous cytokines to see could we in fact measure any elevated endogenous cytokines and what impact did ATI-450 have on those. And in that situation, the cytokines are there in very low amounts, in picograms per mil. And that's a very, very small signal. But we were able to see that ATI-450 had a significant effect on TNF-alpha, IL-6, and some other cytokines as well. So primarily the ex vivo assay is used to assess pharmacodynamic parameters to inform us as far as doses versus pharmacokinetics versus pharmacodynamic response.
Yeah, and I think just one other point on that, as Wally was alluding to, we looked at this multiple different ways. We didn't just kind of pick the best assay and report those results. We looked at IL-1 beta stimulated models, LPS stimulated models, endogenous, exogenous. We've looked at it every which way you can. And I think the comforting thing with it is that it's all confirmatory. And so I think that's in part, what characterizes when we talk about kind of a robust data set. When you look at it through multiple windows or perspectives and you kind of get the same result, that's comforting.
Got it. Going towards the upcoming or the current studies. So the phase 2A RA study used 50 mg BID, ACI 450, and you've dosed up to 120 mg BID with the drug in the healthy volunteer study. Just walk through the rationale behind moving the 50 mg BID dose forward, as well as the 20 mg BID dose that's also being explored in the ongoing RA study.
Right. So as I mentioned before, the 50 milligram BID dose resulted in multiples of the IC80 for inhibiting cytokines at Cmin, at the minimum concentration before the next dose. So what we determined in the multiple ascending dose studies that we could dose higher. We saw no dose-limiting toxicities, but we're already inhibiting the target, as we mentioned, probably over 90% at the 50 milligram dose. And what we really are trying to determine in this current RA study is what's the minimal effective dose. So by dosing down at 20 milligrams, we think we're having significant cytokine coverage over the dosing period. It'll be interesting to know, since we are hitting multiple cytokines, would a lower dose actually even be an active dose as well?
Got it. Can you walk through the safety profile to date with ATI 450, as well as whether anything notable has been seen preclinically through your tox studies?
Well, I'll talk about the preclinical studies, and maybe Neil, you could talk about the clinical studies. But in the preclinical safety assessment, we have completed out the long-term tox out 26 weeks in rodents and 39 weeks in non-rodents. Those data are still coming in. But the 13-week studies were completed to support the RA work in the clinic, and those studies were quite clean. In fact, our highest doses tested in those studies was the no effect level. So there was some mild symptoms, but really no real target organ toxicity was identified in those studies. And it'll be interesting to know with the long-term tox if that's substantiated. But in the clinic, we had a compound was fairly well tolerated. And maybe, Neil, you can talk about it.
Yeah, I mean, I think we saw what you would expect, a few mild to moderate infections, no SAEs with the exception of one patient developed COVID in the post-treatment follow-up period. But so far, I think, as Wally mentioned, it's been pretty well tolerated. We see some transient headache in the early days, which seems to go away with increased dosing. The dizziness that we saw in SAD/MAD work didn't really translate into the RA study. We didn't see any of it. That's something obviously we're gonna follow, but that's also, if it's a real effect, it also seems to be transient and goes away with increased dosing. So far we're happy with the profile, and of course we've got 3 more studies kicked off, and we'll continue to gather safety data.
Great. And so for the 3 ongoing studies with ATI 450, you got HS, RA, and then psoriatic arthritis. Can you just walk through the top line study designs, powering assumptions, and the goal of each of these trials?
Sure. I think the RA obviously is the one dose range. So we have 50 and 20 BID and 80 patients per arm. We're 90% powered on that, and the delta that we're looking to detect there overall is somewhere in the 26 to 30 point range between placebo and an ACR 20. Of course, we're looking at a whole variety of endpoints. It's a pretty much traditional bread and butter RA study design, looking at ACR 50, 70, DAS28 all across the board. Obviously, we want to gather a lot of that information as we plan for what a phase 3 program looks like in that indication. Then psoriatic arthritis, again, pretty much run-of-the-mill study design, similar to what others would embark on in a 2A, 70 patients, so 35 per dose group, and we just have 50 on placebo. And then HS, 35 per dose group, 70 overall patients, 50 versus placebo again. In the HS study, I think the one nuance is that we're looking at all the traditional endpoints, but the primary is a mean change from baseline in the inflammatory nodule count. And we're basically mandating that you have, at least 5 nodules at least 2 sites to be included in the study. And, what we're hoping there is that, when you have more of those, that it kind of washes out, some of this strange placebo effect you sometimes see in these studies. But that's pretty much what we're looking at.
Got it. Okay. And then I saw too you recently raised an additional $75 million a couple of weeks ago. And so with the raise, where does this bring your cash runway to? And then what are your plans with the additional capital?
So what we view the additional capital as operational flexibility. I don't think it's any secret that this market is quite cathartic at the moment. It's in a lot of turmoil. And I think for anybody in our position, particularly my position, you have to be thinking about that macro landscape. And so we had the opportunity, we had some inbound interest and had the opportunity to take additional capital, shore out the balance sheet, gives us a lot of operational flexibility as we move forward. We're now messaging that we get to the end of 25. So, we have really nice cash runway, at least relative to our peers. And we're able to generate quite a number of important data readouts in 23, also in 24 with some of the earlier stage programs. So I think we put the company in a pretty good position. I don't see us doubling down on investment at this stage. I'm taking a little bit more of a defensive approach, just given the tenor of the markets.
Understood. And looking at the rest of the pipeline, so one of your additional assets is a topical soft JAK1/3 inhibitor called ATI 1777, which is being developed for moderate to severe atopic dermatitis. So you reported phase 2 data with the asset last year and are currently kicking off a phase 2B trial in moderate to severe AD this quarter. Can you remind us on the phase 2A data with the asset and how 1777 differentiates versus other JAK inhibitors that are topical?
Sure, Wally, do you just want to mention the top line data and I'll talk about the differentiation?
Yeah, so the study was taken forward into moderate to severe atopic dermatitis patients. The primary endpoint was the change in modified EASI score. But the key top line result that I can convey today is that the EASI 75 parameter for treated individuals was 65%. So quite a robust decrease in that endpoint. The other key aspect of that study was to look at circulating plasma levels of the parent, and they were very low. In fact, almost all samples, over 80% of the samples, were below the limit of quantitation. And those samples were even close to the IC50 for inhibiting JAK enzymes in whole blood. So the hypothesis really played out that we had an impact on the disease with very limited to no systemic exposure.
Yeah, and in terms of differentiation, look, I think if you look across the board with oral JAK inhibitors and topical JAK inhibitors, they work. They're quite potent as anti-inflammatories in a variety of indications. So in terms of efficacy, I think when you look at raw numbers, they're all going to be within points of one another, depending on the patient population. We chose to differentiate based on safety. And, we did that at a base level, selecting against JAK2. I think we've seen that with topical Rux. There's, some warnings on the label about monitoring blood levels, with kind of selecting against JAK2. We're hoping we don't have that concern. We also doubled down on that and went with the soft approach, as Wally mentioned. And we had started that work back in 2016 because we knew that was going to be one of the key things to differentiate topical against topical, is just having that tissue-specific approach. So from our perspective, if you can maintain the efficacy at the site of action and not worry so much about some of the safety issues characteristic of this category, then you're quite a step ahead of the competition from that perspective. We also decided, just from a strategic perspective, to go after moderate to severe disease. We think that's a very good market and a lot more underserved than certainly mild to moderate. And so those are the key items that we hone in on.
All right. Yeah, I guess to your point with Insight's topical ruxolitinib, obviously some language on the label regarding systemic exposure, whereas with your soft JAK, the goal is to not have that. I guess in your conversations with physicians, has that resonated much in terms of that differentiation?
Well, I think it certainly. I mean, I used to practice as a dermatologist. If I don't have to worry about talking about lab, like potentially monitoring labs, and I think that's worse than some of the black box labeling, to be frank, because I think most derms realize that you're probably not going to get levels that are going to have some of those systemic impacts. I think anytime a dermatologist can give a medication to a patient and they don't have to necessarily have in the back of their mind all of those label issues, it's a net positive. And remember, we're treating predominantly young patients, and so you have to talk to the patient, but also the parents. And I can tell you that that's not always the easiest thing. So I think I think that's absolutely something that matters. Safety always kind of matters in topical medicine.
Got it. And one thing too, with the topical ruxolitinib from Insight, so that's a cream-based formulation. Can you just remind us on the planned formulation for ATI 1777 as well?
So we have a spray, and every patient has different nuances in terms of what they like, whether it's cream, gel, spray, ointment. I think we chose a spray because we want to go after moderate to severe disease, and often that has greater body surface area. And, sprays are easy to use. I mean, just think about sunscreens, right? That's all you see anymore at the beach, parents spraying their kids down with sunscreen. There is a convenience factor to that. And so that's kind of why we chose that route. And look, some patients prefer cream. So I don't, I see that as a difference and not necessarily a general differentiating factor. It's more the utility of using it over larger body surface areas.
Okay, and in terms of frequency of use, I know topical Rux is twice daily. I guess what's the intended usage of yours?
So right now we've conducted a study with BID. This next study will explore both BID and QD at the top concentration, which is 2%. So, should that data come out positive on the QD side, I mean, it's always kind of nicer if you can move to a QD dosage form, although in a disease like AD, I don't know that it's a necessity per se. These patients want to get better, and putting something on twice a day really isn't that onerous.
Got it. And turning to ATI 2138, so we expect to have phase 1 data this year with the asset. It's an ITK, TXK, JAK3 inhibitor. Would be great just to hear how you're thinking about the development of this program and what makes it unique versus other I&I drugs out there.
Yeah, so the compound inhibits two key signaling pathways in T cells. So it inhibits both T cell receptor signaling and cytokine signaling that utilize JAK3 in its signaling cascade. So it's a very dramatic immune modulatory effect. Think of it as analogous to cyclosporin, but these targets